Hi, I'm Poe Bronson. I'm the managing director of IndieBio, and this is Founder Stories, where we spend a little time with the CEOs of our graduating startups. And today, I have Dr. Jameel Feshitan. Jameel is the founder and CEO of Advanced Microbubbles. Jameel, thanks for spending a little time with me.
Thank you, Paul. Glad to be here.
I appreciate it. Now, Jameel, um, a lot of people were trying to use diagnostic microbubbles activated by ultrasound as a companion to chemotherapy in hope it could pump the chemo past the blood-brain barrier or tumor barriers. It wasn't working that well for most of them. And then you burst on the scene and you got a lot of attention and you published this data working with NIH. Can you tell us what we're seeing here?
Yeah, thank you, Paul. So this is very interesting data. So just to give you a framework, right? So the blood-brain barrier precludes drugs from getting in, generally 400 Daltons from getting into the brain. So it's a major bottleneck for drug delivery. So the whole point of using ultrasound and bubbles was, in this data set, is to try and use the bubbles to create the temporary passageway for the drugs. Now, NIH NIDA, for the longest time, they were trying to use the conventional diagnostic imaging bubbles. And for years, they were struggling with the issue of consistent dosing of the drugs and basically reproducibility of the results. So what's interesting is, by partnering with us, we solved a problem that took them years in 3 months. So the data you're looking at here is comparing three different mice groups, right? The control, the top image where you inject the drug alone, which has a hard time getting across the blood-brain barrier, so you don't really see much penetration, basically the whites. The middle image is when you use a conventional diagnostic image bubble sizes, and you get some penetration, but it's not that much, and it's also inconsistent because the size is inconsistent. But then the last image is what happens when you use our microbubble technology. And you can see the amount of drug reaching in is at least 10-fold greater than with the conventional bubbles. And it's also homogeneous. So it's something that makes us stand apart. We have the holy grail of bubble engineering, the bubble that is both stable and also uniform in size.
Great. So, Jameel, now off that study, investors might assume you're going into the brain. But in fact, you're doing that through partnerships, and I know a lot of companies are interested in doing that with you. But Advanced Microbubbles itself is going after abdominal cancers, such as neuroblastoma, because it can use conventional ultrasound. So now, during IndieBio, you did some additional data. I want you to tell us what we're seeing here and explain it to us.
Thank you, Paul. So yeah, again, so we're pursuing the abdominal cancer just because it's easier to treat with conventional ultrasound. So the first proof of concept in a mice where we used our technology to try and treat this pediatric cancer disease was performed in collaboration with University of Texas and University of Chicago. So what you're looking at in the graph here is a tumor growth curve when you compare three different groups. The use of a drug alone, a chemotherapy drug, LDOX, the use of LDOX with our technology, and then an untreated control. So what's really interesting in this data is, for the first time, we're able to use a very low dose of chemo, 1 mg per kg, which was able to halt the tumor growth without causing any side effects to the specimen, meaning no weight loss, no formation of cells, or no loss of motor function. That was an effect that was not possible without our technology. As you can see, the chemo only keeps growing exponentially, and the untreated animals also keeps growing exponentially. So it's a very enabling result for clinical translation, especially in this pediatric cancer model.
Great. So now, on behalf of investors, what they want to know and understand is why your bubbles are different from everyone else, both on a performance and science point of view, but really from an investor point of view, we need to know, of course, you have the patent, obviously, but why can nobody imitate the properties of your bubbles? Teach us why you stand out.
That's an excellent question, Paul. So this microbubble technology is something I've been working on for over 13 years. And the method of making this bubble is something I've been working on since my time back at Columbia University. And it's a very involved process. It's not a simple press one button. It requires a lot of knowledge of the key trade secrets and methodologies involved. So it's about 17 different steps involved in the process of making the bubbles, making them reliably, and making them at scale. So it's something that we are very specialized in that, and people have been trying and they haven't really had that time, been able to figure it out. So that's why they reach out to us, a small startup company.
Thank you. Now, investors want to know your reg path and what this means for future clinical trial sizes and the kind of expense going into your company. Can you articulate on that, please?
That's also a very excellent question, Paul. So we specifically are working on a neuroblastoma model because one of the advantages of that is that it is a rare disease. So, well, the two advantages are our co-founder is also an expert on treating the disease, but the main advantage for us in terms of the next steps for our company is shortening our time to the market. So there's this law called the Rare Disease Act or Orphan Disease Act, where drugs that are approved to treat or that are meant to treat rare diseases get a much shorter timeline to the market. So neuroblastoma, which has less than 1,000 patients, US patients per year, it's such a disease where if you can, develop a drug to treat this disease, the FDA will give you a lot of benefits like R&D tax benefits. You cut down the amount of money and expense you need to get your drug approved, and also the amount of patients needed for approval. You can get approval with as low as 8 patient numbers, and you save up to $2.5 million in terms of cost. And then in terms of time, so you can also save the time to get to clinics from typical time of like 10 years for most drugs to down to almost 3 to 4 years. So it's really advantageous for us to be able to start out with this rare disease pathway and then expand to other cancer indications.
Yeah, an 8-patient trial and the relatively lower expense of that, which should be appealing. Now, the big market here is the pancreatic cancer, and and I know that after IndieBio, you're going into models on pancreatic cancer. Can you share who your partners are?
Yeah, so we're following up. We plan to follow up the study in pancreatic cancer, as you mentioned, and we are very lucky to be able to work with our current collaborators at the University of Texas, but we'll also be starting a collaboration with the University with New Mexico Tech. So there's a group that's developing novel drugs to treat pancreatic cancer. So we're going to work on using our technology to enable the safety and efficacy of their drugs in a pancreatic cancer model, and that's we're hoping that will be part of our in vivo data package that will enable us to get into the clinics.
Great. Jameel, thank you for spending a little time with me today.
It was a pleasure, Paul. Thank you.